DNA and RNA are known as nucleic acids, which are very important molecules for all living organisms because they carry and use genetic information. Although DNA and RNA are closely related, their structure are different, and this difference allows them to perform different functions in the cell. First, we will focus on DNA, which stands for deoxyribonucleic acid. DNA has a very unique structure called a double helix. This structure looks like a twisted ladder, where the side of the ladder form the backbone, and the rungs are made of base pair. DNA is made up of many repeating units called nucleotides, and each nucleotide has three main components, which are deoxyribose sugar, phosphate group, and nitrogenous base. There are four types of nitrogenous base found in DNA, which are adenine, thymine, guanine, and cytosine. In DNA, these bases follow a specific base pairing rule, where the adenine will always pair up with thymine, and meanwhile the cytosine will always pair up with guanine. These base pairs are connected by hydrogen bond, which helps stabilize the structure of DNA. Next, we will focus on RNA, which stands for ribonucleic acid. RNA has a simpler structure compared to DNA. Unlike DNA, RNA is usually single-stranded, meaning it has only one strand instead of two. Similar to DNA, RNA is also made up of nucleotides, and each nucleotide consists of ribose sugar, phosphate group, and nitrogenous base. However, RNA has a slightly different set of bases, which are adenine, uracil, guanine, and cytosine. In RNA, uracil will replace the thymine, which is found in DNA. When base pairing occurs in RNA, adenine will pair up with uracil, and meanwhile the guanine always pair up with cytosine. Because RNA is single-stranded, it is less stable than DNA. However, this makes RNA more flexible, allowing RNA to exist in different forms, such as mRNA, tRNA, and rRNA, and each type of this has a specific function related to its structure.
Build up or break down items they add on. So here we
Hi Iza. Now I finished to watch my video, but I still can't understand what the DNA function actually. Do you know?
Yeah, let me explain you about DNA function. Because ribonucleic acid or DNA function is to carry genes, the information that specifies all the protein that make up an organism. As every organism contains many genes in its DNA, different types of proteins can be formed. DNA also transmitting hereditary information from parents to offspring through replication and gene expression. Replication process is to transfer the genetic information from one cell to its daughter cell during cell division. DNA also controls protein production. DNA directs all cellular activities by controlling which proteins are made and when through gene regulation.
There are three main types of RNA involved in protein synthesis. They are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Transcription is the process of RNA formation from DNA. First, mRNA. mRNA is transcribed from DNA and contains the genetic blueprint to make proteins. It carries genetic message from DNA in nucleus to ribosome, which is the site of protein production. Researchers are also studying mRNA as an anti-cancer treatment due to its ability to modify cells. tRNA. tRNA is RNA molecules that translate mRNA into protein. The primary function of a tRNA is to carry amino acid on its 3' acceptor site to a ribosome complex with help of amino acid tRNA single rRNA. rRNA forms ribosomes, which are essential in protein synthesis. A ribosome contains large and small ribosomal segments. The ribosome contains an exit E, peptidal P, and acceptor A site to bind amino acid RNA and link amino acid together to create polypeptides. In summary, mRNA carries the message. tRNA delivers the building block, and rRNA builds the protein. Together, these RNA types are essential for protein synthesis and life.
Helices bind to the origin and unwind the DNA double-stranded helix by breaking the hydrogen bond with the complementary strand at the replication fork. So now each strand serves as a template. Since we don't want this strand to rejoin back, so here comes single-stranded binding protein (SSBP) that will bind to separated DNA strand and prevent them from rejoining back. Topoisomers bind and relieve overwinding strand due to the supercoiling of DNA molecule by breaking, swiveling, and rejoining DNA strand. Primers catalyze the synthesis of RNA primer by adding RNA nucleotide which complement to the DNA template strand. This is really important because otherwise DNA polymerase won't know where to start the DNA synthesis. Here's come DNA polymerase III bind and catalyze synthesis of new DNA strand by adding DNA nucleotide to the RNA primer, which is complementary to the DNA template and elongate in 5' to 3' direction. The DNA polymerase III build a new DNA strand by adding one RNA primer in the 5' to 3 direction, and it synthesizes continuously toward the replication fork. This is called the leading strand. At the same time, DNA polymerase III also build new DNA strand by adding more than one RNA primer that synthesizes discontinuously away from replication fork from 5' to 3' direction, known as a lagging strand. This process will be for forming a series of short fragments called Okazaki fragments. DNA polymerase I removes RNA primer and replace it with the DNA nucleotide. DNA ligase catalyzes the formation of a phosphodiester bond between the two Okazaki fragments.
Right now, I'm going to compare eukaryotic DNA replication and prokaryotic DNA replication. First, let's talk about their similarities. In both eukaryotic and prokaryotic cells, DNA replication is semi-conservative, meaning each new DNA molecule contains one original strand and one newly synthesized strand. In addition, replication in both systems also occurs in the 5' to 3' direction. Then, both use same enzymes such as DNA helicase to unwind the DNA. Next, they also use single-stranded DNA binding protein to stabilize the unwound DNA strand in both processes. Then, they also use topoisomerase that will bind at the region ahead of the replication fork to prevent supercoiling. Next, both prokaryote and eukaryote also use primase to synthesize RNA primer and DNA ligase to seal Okazaki fragment. Now, let's look at the differences. One major difference is the location. In prokaryotes, DNA replication occurs in the cytoplasm because prokaryotic cells do not have a nucleus, while in eukaryotic cells, replication takes place inside the nucleus. Another key difference is the structure of the DNA. Prokaryotic DNA is delta and usually has one origin of replication, while in eukaryotic DNA is linear and has multiple origins of replication, which allow large genome to be copied more quickly. Next, they also differ in origin of replication. For prokaryotes, they have one origin of replication per DNA molecule, thus the replication of DNA occurs at one point in each prokaryotic DNA molecule, while for eukaryotes, they have many over 1,000 origins in each eukaryotic chromosome, thus replication of DNA occurs at several points simultaneously in each chromosome. Then, they also differ in recognition of origin. In prokaryotes, the origin of replication is recognized by DNA A and DNA B, while for eukaryotes, it is recognized by pre-replication protein complex. Next, they also differ in synthesis of new DNA strands. For eukaryotes, they involve 3 DNA polymerase. First, DNA polymerase alpha. The function is to initiate synthesis of new DNA on both leading and lagging. Then, DNA polymerase delta. It is to complete synthesis of Okazaki fragments. And lastly, DNA polymerase epsilon. It is to complete the synthesis of leading strand. Proceed to prokaryotes. It only involves one DNA polymerase. It is DNA polymerase III. The function is to synthesize new DNA on both leading and lagging strand. Next, excision of RNA primer. During removal of the RNA primer, eukaryote use flap endonuclease 1 and RNase phage to remove the primer, and DNA polymerase alpha to replace it with DNA, whereas prokaryote use DNA polymerase 1 to remove the RNA primer and fill the gap with deoxynucleotides. Then, proofreading of new DNA strands. For proofreading, eukaryotes rely on DNA polymerase delta and epsilon with 3' to 5' exonuclease activity to correct errors, while prokaryotes use DNA polymerase 1 and 3 to proofread and fix mistakes during DNA synthesis.